Okay, so right now what we're going to do is page 17 in the notebook. So let's go over basically right now proteins. We just talked about DNA, a review a little bit. Now we're going to talk about the role of proteins, what makes proteins different from each other, and a little bit of review on amino acids. Okay, so why do we need proteins? Well, they make up your enzymes to digest your food. Hemoglobin, which we know is necessary to get oxygen to our body cells. Makes your hair and nails. It's a substance called keratin. It's also what's found in the baleen of whales and hooves and things like that. So it's just like that hard material right there. It creates, or it is a hormone. So they're hormones for cell-to-cell communication. That's how cells talk to each other hormonally. Very important. Clots your blood. So every time you bleed, you can stop the bleeding. And then makes antibodies. Antibodies are what fights the infections and germs in your body. They destroy germs. Okay, so that is what proteins do. So basically, if you didn't have proteins, you couldn't eat. It would be pointless to breathe because you couldn't bring it to your body cells. You'd have no hair or fingernails. Your cells wouldn't communicate with each other. You would bleed to death and you would always be sick. So what's the purpose of this? Proteins are important. Okay, all proteins are different from each other. They all shop from the same amino acid store. So they're all made out of the same 20 building blocks, but just in different arrangements and sequences. So they're different from each other in the amount of amino acids used. One of them has like 15,000 amino acids in it. The type of amino acids used. And the sequence, like what is the order of amino acids used in the polypeptide chain, as well as the structure of the protein. Like you can have square proteins like hemoglobin that has the 4 irons in the corner, or globular proteins. It just depends on the protein. Okay, so I want you to think of a protein like this. It's basically a bead necklace. It's not a protein yet though until it's folded. That's what makes it a protein, because we talked about this in first semester. When you unfold a protein, you call it denaturing. So you unfold a protein with pH and temperature. So this is just a polypeptide. All of these beads represent amino acids, and then they fold in on each other like that, and now it's a protein. Okay, so here we go. All proteins have the same set of, or use the same set of 20 amino acids. So the amino acids, remember, are the monomers, whereas the whole protein itself is the polymer, but in different amounts and sequences. The 20 amino acids all have the same basic structure, but differ in what their R group is. Remember I told you the R group right here, it's like beads on a chain, or like a charm bracelet. Everyone has, hi Juju, everyone has a different R group. Okay, so the same background is this nitrogen, carbon, carbon background. So if you look at all 20, if you were to count them, there'd be 20. If you look at the NCC backbone, they all have these same basic features, but they all differ in this. So for every single one of this is the R group. R is just a variable like X. It doesn't mean anything. Okay? And so when different amino acids come together, it's this process right here called condensation. We talked about this last semester. Basically, the hydroxide from amino acid number 1 comes together with a hydrogen from amino acid number 2. They come together, out goes the water. The water is either urinated or sweated, etc. And now the 2 amino acids are linked together. Here's 1 and here's 2. And that bond right there is called a peptide bond. Tomorrow what we're going to go over is how your ATCG sequences code for which amino acid to bring in. Let's actually look at that for a second right here just to give this more context. Go here. This scientist came up with the codon wheel. Move this down here. It was a really amazing contribution to science. Let's see. Okay, I'll teach you more in depth on how to use this tomorrow. But basically, you have 4 letters in your DNA alphabet, A, T, C, G. And there's only 64 possible arrangements of those 4 letters if you did the statistics behind that. And the scientists figured out what the code was for each amino acid. So every set of 3 letters codes for an amino acid. It's called a codon. So like, let's say in my DNA, I had the sequence CCC. Well, my mRNA would have the opposite on it, which would be GGG. And so when my ribosomes read it, it would read G in the first letter, G in the second letter, G in the third letter. I need to bring the amino acid glycine in. So this codon wheel interprets the ATCGs of your DNA into the amino acid sequences of your proteins. It's pretty cool. We'll go into that tomorrow. But that's all for now. This is what proteins do. This is how they're different from each other. Remember the 20 amino acids that all proteins shot from. They're all the same except for what they have right there in place of R. And when they come together, they form a peptide bond.